Ever wondered how your body makes proteins? Today we'll learn the story of protein synthesis inside our cells. The first step of protein synthesis is transcription. Inside the nucleus of the cell, an enzyme called RNA polymerase unzips a section of DNA. This results in two strands, the upper strand, known as sense strand, and the lower strand, known as antisense or template strand. The first step is initiation. It begins at the promoter region, where it tells the RNA polymerase where to begin transcribing. Now, elongation can begin. The RNA polymerase will start reading the antisense strand in the 3 to 5 direction, adding complementary RNA nucleotides to the three ends of the RNA strand one by one, forming a growing mRNA strand. This process will end once the RNA polymerase reaches the stop point, which is the terminator. This process of ending transcription is called termination. After transcription, the newly formed pre-mRNA undergoes editing to become mature mRNA. This includes splicing, which is cutting out unnecessary parts called introns by spliceosomes, and joining the remaining exons together. Next is capping, which is adding a special cap, GTP at the 5 end and poly A tail at the 3 end. The cap and tail helps mRNA find and attach to ribosomes during translation and protects it from enzymes that break it down. Now, the mature mRNA leaves the nucleus and travels to the cytoplasm, where the ribosomes are located. We now proceed to translation. It begins with initiation, where the mRNA binds with a small ribosomal subunit upstream of the start codon, which is AUG. A special initiator tRNA molecule carrying the amino acid methionine binds to the start codon on the mRNA. This triggers the large ribosomal subunit to bind, forming translation complex. The initiation process is completed here. We now proceed to elongation. During elongation, tRNA molecules bring specific amino acids to the ribosome. Inside the ribosome, the mRNA is read codon by codon. The ribosome has 3 sites, the A site, P site, and E site. Each anticodon of tRNA pairs with the complementary mRNA codon. A charged tRNA molecule binds to the A site, and a peptide bond forms between its amino acid and the one at the P site. The ribosome then moves along the mRNA, sliding one codon to the right. The uncharged tRNA exits from the E site, and the A site is ready to accept the next charged tRNA molecule. The ribosome continues linking these amino acids together using peptide bonds, forming a growing chain called a polypeptide. Translation ends when the ribosome encounters a stop codon on the mRNA, which could be UAA, UAG, or UGA. A release factor binds to the A site at the stop codon, causing the completed polypeptide chain to be released from the tRNA and the P site. The released polypeptide chain then folds into its functional 3-dimensional structure, becoming a functional protein.